Hello viewers, welcome to my channel Chem Corona. In this video, we are going to discuss SAR of fluoroquinolone antibacterial agents. The general structure of fluoroquinolone is as shown here. It consists of a quinoline ring. If we want to number the quinoline ring, we start with the nitrogen atom. It gets position 1, 2, 3, 4, 5, 6, 7 and 8, where X can be either carbon or nitrogen atom, and R1 is fluorine in most of the fluoroquinolone drugs, or it can be either aryl or alkyl moiety, whereas R7 can also be either alkyl or aryl moiety. Now let us see the different structural sites which determine the antibacterial activity of fluoroquinolones. The nitrogen atom is responsible for the overall potency of the drug. The carboxylic acid at third position and the oxo group at fourth position, they are responsible for binding of the drug. The fluorine atom at sixth position is responsible for cell penetration and Gyrase affinity, whereas the R7 substitution is responsible for the overall antibacterial spectrum, and X can be either carbon or nitrogen, and it is responsible for anti-anaerobic activity. The different nucleus which is present in fluoroquinolones are: most of the drugs they contain 1,4-dihydroquinoline ring. In some drugs, quinoline ring is also present. Example is siloxacin. In quinoline, the nitrogen is present at 1 and 2 position, whereas some of the drugs they also contains 1,8-naphthyridine ring, wherein the nitrogen is present at 1 and 8 position. Example is nalidixic acid and inoxacin. Now let us see the structure-activity relationship of fluoroquinolones. Substitution at N1 position. Earlier, ethyl molecule or group of smaller size is considered best for the activity. Introduction of cyclopropyl group is found to be extremely potent. We can also have tertiary butyl group at N1 that increases the activity against gram-positive bacteria, whereas N1 fluorophenyl group, it improves the activity against anaerobic pathogens. Let us see the drugs. Norfloxacin, it contains ethyl group at N1 position, whereas ciprofloxacin, it contains cyclopropyl group, and tosufloxacin contains 2,6-difluorophenyl group. Substitution at C2. Replacement of the hydrogen, it decreases the activity, whereas sulfur atom can be tolerated if it is a part of an additional ring system at the second position. Substitution at C3. Carboxylic acid moiety is required for binding of the drug to the DNA gyrase. We can also introduce other acidic groups like sulfonic acid or phosphonic acid, but it leads to loss of activity. Incorporation of aldehyde or carboxylate esters can be afforded at third position. Substitution at fourth position. The oxo group at fourth position is essential for antibacterial activity. If we replace this oxo group with thiooxo or sulfonyl group, it results in loss of activity. Substitution at C5 position. At fifth position, amino group enhances the absorption and distribution of the drug. Example, sparfloxacin. We can also have alkyl groups larger than methyl groups at fifth position, but that decreases the activity. Substitution at C6 position. Fluorine atom causes drastic increase in the activity. Fluorine group is responsible for DNA gyrase, binding and cell penetration. Other groups like chlorine, bromine, methyl, they are active at 6 position, but they are less potent than fluorine group. Substitution at C7 position. Piperazonyl group with fluorine atom at C6 position is found to be potent and active against gram-positive and gram-negative bacteria. Small groups like methyl, amino have moderate to weak activity. Substituted piperazonyl at 7th position enhances the activity against gram-positive bacteria, whereas other substitutions like piperazonyl, pyrrolidinil or morpholinil can be afforded at 7th position. Substitution at C8 position. C8 contains either fluorine or chlorine, and it is found to be more active in vivo and has better oral absorption. At 8th position, methoxy group increases the activity, specifically against anaerobes. Fluorine at 8th position is responsible for producing phototoxicity. Example is sparfloxacin. We can also have ring fusions at 1 and 8 position. Example is ofloxacin, which is a second generation fluoroquinolone, and levofloxacin, which is a third generation fluoroquinolone. We can see here this 1 and 8 position, they are fused. So this is called as ring fusion at 1 and 8 position, which introduces a stereocenter which leads to active molecule like levofloxacin, wherein the levo isomer is more active compared to the ofloxacin. Similarly, ring fusion can also be there at position C6 and C7. Example is sinoxacin, which belongs to the first generation fluoroquinolone. To summarize the structure-activity relationship of fluoroquinolone, the N1 substitution is responsible for potency and has some effect on pharmacokinetics, whereas the C3 and C4 are responsible for DNA gyrase binding activity. Substitution at 5th position, it controls the potency and adds towards the activity against gram-positive bacteria. Fluorine atom at 6th position is responsible for cell penetration and gyrase affinity. The substitution at 7th position, it controls the overall spectrum of the drug and also the pharmacokinetics, whereas C8 substitution controls the pharmacokinetic and also increases the activity against anaerobes. So thank you for patient listening and happy learning. Like, share and subscribe my channel Chem Corona.